This is Yannis Manis here to talk about the gram stain procedure, its steps, and their importance to the gram stain procedure. Step 1.
Obtain a clean slide. Use your loop and water bottle to place 3 drops on the slide. The first drop will be used to make a smear of E. coli. The third drop will be the S. aureus, and the center drop will be the patient samples.
This step is basically preparing the slide to be smeared. We obtain a clean slide in order to prevent contamination from a previous slide. We use a loop in order to have circular blots for our smears to make things more examinable. And then we use the water to have a place for our smear to be placed.
Aseptically remove a sample of E. coli. Mix the inoculum into the first drop. Spread the drops to create a thin smear.
Step 2. We aseptically remove the sample of E. coli to prevent contamination again, and then we spread it onto our water circle to prepare the smear. Step 3.
After flaming your loop, aseptically remove a sample of S. aureus. Mix the S. aureus into the third drop of water. Spread the drops to create a thin smear. Last, add patient sample to the center drop in the same fashion.
It's basically the same thing as step 2, except with our remaining 2 samples. Step 4.
Air dry the slide.
Air drying the slide prevents the liquid and bacteria from coming off of it and possibly losing the sample. Step 5.
Heat fix the slide. Place the heat-fixed slide on the staining tray.
We heat fix the slide in order to fix the bacteria onto the slide. This basically kills the bacteria so they can no longer move. Step 6.
Cover the smears with crystal violet, the primary stain. Stain for 1 minute.
The primary stain, which is crystal violet, is used to stain our bacteria so they can be seen under a microscope. And step 7.
After 1 minute, tip the slide and gently wash the slide with distilled water.
Cleans off any excess stains so the bacteria can be seen under the microscope. Excess stain would just get in the way and mess up the results. Step 8.
Cover the smears with grams iodine, the mordant, for 1 minute.
The iodine causes the primary stain to bind to the peptidoglycan layer of the cell wall. And since gram-positive cells have this layer, it is bound to it. But since gram-negative cells do not have a very thick layer of this, it is not bound to it.
After 1 minute, tip the slide and drip the decolorizer acetone alcohol down the slide. Decolorize for 15 to 20 seconds. The decolorizer dripping off of the slide will initially be very dark blue. After 15 to 20 seconds, the decolorizer color will be much lighter, but still slightly blue.
The decolorizer gets rid of the primary stain that was found in the cells, but not the primary stain that was found in the cell membrane, which means that the gram-negative cells now are colorless and the gram-positive cells still have their color.
Cover the smears with safranine, the counter stain, and stain for 1 minute.
Step 10. The safranin, which is the counter stain, stains the remaining colorless bacteria. Since gram-negative cells are colorless, they will be dyed a light pink. Step 11.
Rinse the slide with distilled water and blot dry.
We rinse the slide with distilled water to get rid of any excess safranin or other stain that was on the slide, and we blot it dry to get rid of any liquid found on it. And then step 12.
Using oil immersion. Gram-positive cells will appear purple and gram-negative cells will stain pink.
We use oil immersion in order to see our cells and have a result for our gram stain test. Thank you. Good night.